Greetings, YouTube! My name is Dana Brimmer, and I'm coming to you today in my capacity as an organic chemistry student and patent nerd over at the University of California, Davis. That's almost there, it's there. Today we'll be learning about carboxylic acid derivatives. But not just any carboxylic acid derivatives. Today we'll be learning specifically about esters. Before we get going, though, let's do a little bit of review. What are carboxylic acids? Well, carboxylic acids are molecules with a carboxy group. That's this part here in the middle. And a carboxy group is basically a hydroxy unit attached to a carbonyl carbon. That's here. In nature, we often find complex organic systems that combine these carboxy-carbonyl groups with, well, something else. That something else is called a heteroatom, or a non-carbon atom, and it's often part of another functional group. And collectively, we call these carboxylic acid derivatives. Typically, they're written like so, where we replace the hydroxy unit with an L, Y and L, because these groups often act as good leaving groups. Your organic chemistry book often focuses on four major types of carboxylic acid derivatives: the halides, anhydrides, esters, and amides. This video is specifically about esters. Esters are super important in nature. This video introduces several of these common types of natural esters with a brief description of their biological functions. The first type of ester we will discuss are waxes. Waxes are esters that are made up of long-chain carboxylic acids and long-chain alcohols. How long? This is a formula of a typical example of a wax. There's 1, 2, 3, 4, 5, 6, 25 carbons on the left side, and 1, 2, 3, 4, 5, 6, 7, 8, 29, 30 carbons on the other side. This is a long chain carbon, giving this wax a hydrophobic property. Now, as a reminder, hydrophobicity is a super important chemical property in esters for reasons that we'll get into below, but for now remember that it's basically a molecule's physical property of being seemingly repelled by water. Although strictly speaking there is no repulsive force involved, it's rather an absence of an attraction. The sample above is beeswax, and it's an example of how waxes are often used in nature to form hydrophobic and insulating coatings. The specific uses in nature that are common are on skin and fur of animals, the feathers of birds, and the fruits and leaves of many plants. Waxes are simple esters, but fats and oils are more complex types of esters. Fats and oils, in fact, are triesters. That's 1, 2, 3 esters derived from glycerol with a long chain carboxylic acids attached. These are also known as triglycerides. On the right, you'll see the long chain carboxylic acids, which are also known as fatty acids. Fatty acids are typically unbranched and contain an even number of carbon atoms. If they're unsaturated, like this one above, double bonds are usually cis. And just as a reminder, unsaturated just means that there are some double bonds. Fats, in particular, serve in nature as biological energy reserve. That is energy that is stored in tissues until your metabolism transforms the energy into CO2 and water. This is the approximate function of oil in plant seeds. Now, we've learned about waxes, fats, and oils, and often these are lumped together into a category known as lipids. What are lipids? Well, they're just low-polarity biomolecules. That means that they're soluble in nonpolar solvents, and this property is a key to many of their biological functions. Remember those hydrophobic tails above? That's what gives them this low-polarity attribute. One particularly important type of lipid is a phospholipid, which is derived from carboxylic acids and phosphoric acids. They contain two long hydrophobic fatty acid chains and a polar head group, the phosphate or choline substituent. The structure of the phospholipid enables them to form micelles in an aqueous solution. Basically, the heads are attracted to water, but the tails are hydrophobic. So how do you make the whole phospholipid happy once it's in polar water? One way is to arrange the heads so that they create a barrier and separate the tails from the water, the polar water. And so the heads will be arranged in a ball with all the tails pointing inside in a hydrophobic space. You can imagine more complex arrangements than a micelle, and indeed the phosphoglycerides are also commonly arranged in a sheet known as the lipid bilayer. Basically, instead of a ball, a sheet is made where there are two layers of polar heads, creating an interior non-polar space. And these sheets can then also be arranged into a ball, creating a barrier between a polar substance on the outside of the ball and then an interior space, the very interior, which can also be polar. This is actually a relatively stable molecular assembly, and it has a distinct advantage over micelles in that it's not so limited in size. Why do we care? Why is this important? Well, the ability to separate the interior space from the exterior space with a permeable barrier, which can allow for molecular transport into and out of the interior of the ball, is actually what makes cell membranes work in nature. And in fact, the development of the cell membrane has been hypothesized to believe a key step in the evolution of life on this planet.